So that's a great question. Why can't viruses replicate on their own? Why do they need a host cell? Well, viruses aren't technically alive, and they're classified that way mostly because they can't replicate on their own. But when you really get down to it, they're just a bunch of molecules. They are, when you really break it down, a strand of DNA or RNA. The Ebola virus is RNA, and it's single stranded, encased by what's called capsin, or a protein capsule that encloses the RNA. And then there's a membrane that surrounds it. And it has glycoproteins, which are basically like receptors, you can think of them as. So, when the Ebola virus interacts with your cell, first the glycoproteins bind to receptors that are on your cell's membrane. These receptors tell the cell basically that it's okay for the virus to come in. So the viral membrane merges with your cell's membrane and they kind of become one. It adds to your cell's membrane and makes it a little bigger, which your cell wants because your cell wants to be bigger. It wants a larger membrane. That's just kind of how your cell likes it to be. So the virus comes in, it binds with your cell's membrane, and then it releases the capsid into your cell. So this capsid, which contains the RNA or DNA, in the Ebola virus case, RNA, is released into your cell, and the capsin will kind of break down inside of the cytosol of your cell. The capsin will release the RNA material into your cell, and on this RNA strand is polymerase, RNA polymerase, and it will transcribe the RNA into mRNA. So, once it is transcribed into mRNA, it can be translated by ribosomes that are inside your cell. So the ribosomes don't know any better than to just translate this RNA. They just see it as another piece of mRNA that needs to be translated. So the mRNA is translated into the protein structure that encases the virus, that basically encompasses the entire virus. It translates all the proteins that are needed to create a new virus. While this is happening, the RNA polymerase also replicates the RNA. So it creates a bunch of new strands of RNA, which are going to be put into the new virus. Once this all happens, and you have your proteins that are going to become the new virus, and you have your RNA that's going to be inside the new virus, it gets transported to the endoplasmic reticulum, where it's packaged into a vesicle and kind of takes shape into the new protein or into the new virus that it's going to become. It kind of takes shape, it's packaged in this vesicle, and then it's shipped out to the Golgi apparatus, where it is processed, it's kind of moved through the Golgi apparatus, and then it reaches the cell's membrane. Again, and this time it's a new virus. It is now a fully replicated and newly formed virus that has been reproduced by your cell's own functions. So, once it gets here, what happens? Well, in the case of Ebola virus, it goes through a process called budding, in which instead of having its own membrane that it creates, it actually merges with the cell's membrane and pushes out and kind of creates a bubble. So here's your cell's membrane, and it kind of pushes its way out and envelops itself in this new membrane from the cell. So it goes ahead and just steals your cell's membrane and wraps it around itself. This is now the membrane of the virus. It has stolen part of your cell, and it is now a new, fully formed, membrane wrapped and enveloped Ebola virus. That's the full process of how Ebola virus is able to replicate without its own processes. I think it's really interesting. And I think that the fact that it wraps itself in your cell's own membrane probably leads to it replicating even faster because it's able to bind with other cells easier because it's wrapped in the cell's natural membrane. I think that's all really interesting, and I think that it adds to part of the reason that we should study viruses and that viruses are so cool.